Master in Anatomy and Physiology with Indramani Pandey.
Guys, this is my another lecture regarding cell membrane, which is very important and the basic component of eukaryotic cells. You may have membrane in the case of prokaryotic cells also, but the cell membrane is a very unique component of the cell. And in last lecture, we talked about the nucleus, which is a master organelle of the cell, which has genetic information and which usually controls various type of functions, right? So we need to know the general concept of nucleus also. So in today's lecture, we're basically going to focus on the structure of the cell membrane. What are the main components, the ingredients of the cell membrane, and its nature, and how the molecules travels from inside compartment to the outside compartment in the cell. We're going to focus on those. Now let me start with the cell membrane. The cell membrane is a very unique structure which is semi-permeable, or which is selectively permeable, which is not open for all types of substances. Okay, there may be good substances, there may be bad substances. You need to show your ID to enter the cell, right? So if you enter the cell, you can do different things. There are like too many entertainments right there. So to get that entertainments, you may pay, like buying tickets. You know what I mean? Like to go Six Flags, you're gonna buy some tickets, right? Like same type of thing. To go fun park or those things, right? You have to buy tickets. Same way, you have to show ID, and you have to work together with other types of friends or structure so you can go inside the cell. So it is not easy just come and just go inside the cell because of the nature of the cell membrane. Okay, so let me tell you the structure of the cell membrane. Let me start with that. So if you look the cell membrane, usually most of the time the cell membrane is made up of the phospholipid. What is a phospholipid? Phospholipid has phosphate, glycerol, and the fatty acid on it. So that's why it is called a phospholipid. If you think of phospholipid, phospholipid has hydrophobic and the hydrophilic region. The hydrophilic region, what I mean by that is it loves water, or it is polar in nature. And the hydrophobic means it does not like water, or it is non-polar in nature. The lipid soluble, any type of substance like lipid soluble, they can diffuse through the membrane. But what about other type of substance that are not lipid soluble? You need channels. You need some type of structure so you can get in, right? So let me tell you one thing. As I told you, the head region is a hydrophilic in nature, but the tail regions are hydrophobic in nature. And remember that these hydrophobic and the hydrophilic region, okay, makes it selectively permeable. Okay, so anything cannot like go inside. I need to go inside, so you cannot go inside. You need permission, okay? So that permission is through some channels like integral proteins and through peripheral protein. We're gonna discuss those in brief in few moments. So let me tell you that in the case of the phospholipid bilayer, here on the tail region, you may see some type of bending here. Some chains are straight and some chains are bending. That is called a kink. That means it is an unsaturated fatty acid. If there is a kink, that means there is a double bond. It is an unsaturated fatty acid. Okay, you need to understand. I zoom in to one phospholipid and I take it out that phospholipid and I see the structure. I saw that the head region has phosphate group and the middle part has glycerol and the bottom part has a fatty acid which may be saturated or which may be unsaturated. The membrane, the stability is basically depends on the hydrophilic hydrophobic region, not the hydrophilic, hydrophobic region. And in between those states, we have some oval shaped structure here that is called a cholesterol. Cholesterol has a great function in the permeability and it stabilizes the membrane. So that's the best and that's the main idea you need to understand. And the thing is, cholesterol, usually at a moderate temperature, it makes this layer of phospholipid bilayer less fluid, or less fluidity will occur in the moderate temperature. When the temperature is low, what will happen? Okay, what will happen? It hinders the solidification. Okay, so that's the main important function of the cholesterol right there. Cholesterol is not always bad. Okay, so that's why cholesterol is also an important component of the what? Cell membrane. You may have protein, you may have different types of carbohydrates. You're definitely gonna have proteins. Okay, these proteins gonna cooperate with each other and do different things. For example, glycoproteins. What they do is they play an important role in the what cellular recognition and in the immune responses. And what they do is they help to stabilize the membrane structure. Okay, adhesion between the cells. That's the main function of glycoprotein. What about carbohydrates? Carbohydrates, they are found on the outer layer. Okay, you cannot find in the inner layer. Okay, you can only find on the outer layer. They're linked to producing the protein. Okay, that's the main important thing. Okay, like a glycoprotein. Okay, some proteins, okay, we call them as a peripheral protein, as I told you, that is stuck to the surface of the membrane. They are not across like a integral protein. They're gonna come and sit on the surface. Okay, they are loosely bonded, but the integral proteins are very tightly bonded. If you're changing the pH, you can change the peripheral proteins easily. Okay, you can mess up with the peripheral protein, but the integral protein, okay, you need little more energy to change that. So integral proteins are more stable than the peripheral protein, that we need to understand. The glycolipids act as surface receptors and they stabilize the membrane, common in like a brain cells and the nerve cells. You may have that. Okay, that's the important thing you need to understand. The outer membrane and the inner membrane are slightly different from each other due to the presence of the component, the inner component. Okay, the component of the outer component of the cell membrane, usually those are phosphatidylcholine, okay, and sphingomyelin. Those are the outer components usually. Okay, the inner components are usually have phosphatidyl phenylamine and you have a phosphatidyl serine. These are the usually these are the components. Okay, you can see the name here. These are usually very difficult name, but what I want you to know is these layers are slightly different. Okay, due to the presence and the absence of these types of component. Usually found outer component, you find this. Inner component, you find this usually. Okay, and you may not find these components on the outer part and outer ones on the inner part. So that's the main thing we need to understand here. Okay, so as I told you, the integral proteins are very important and they have water filled here. If something is like water soluble, like ions, sodium, potassium, calcium, they can go through there. Okay, that type of channels, usually the water passing channels, usually called aquaporins. Okay, and when these ions go inside and outside, those transportation may be active, passive, depends on like different types of things, right? So we need to understand that. What will happen is these channels work together, as I told you, right? Sometime, uh, sometime you may have receptor cells here. Okay, the receptor cells, what they do is these receptor cells, they will bind to something. Okay, these receptor cells will bind to something, and they may help in the transduction of the signal. Okay, that's the very important component we need to understand. Okay, sometime you may do like cell-to-cell recognition. The cell-to-cell recognition, like for example, some type of glycoproteins, as I told you, they serve as identification. Like a tags, identification tags. Okay, that are specifically, what recognized by the membrane proteins of the other cells. That type of cell-cell binding is usually what short-lived, but it will happen. Okay, sometime what will happen is you may have like a structure which are running from top to the bottom, which may connect the cytoskeleton to the outer component. For example, collagen from the outer extracellular matrix to the inner cytoskeleton, let's say collagen and any type of other type of molecule, right? So they can work together, and they will help the cell membrane stabilization also. That's a very important thing we need to understand right there. That we call attachment to the cytoskeleton and the extracellular matrix is a very important thing we need to understand. So anyhow, these peripheral proteins also working hard, okay, for the binding site. As a binding site, you may have enzymes over there also, right? Enzymatic things also going on here. They may change the conformation of it, and they can trigger the inside compartment to change its like structure or produce new things. Okay, so those enzymes may work there too. The peripheral proteins usually they are the part of the cytoskeleton, inside one. Okay, whatever proteins you find in the cytoskeleton usually are the peripheral ones. Okay, that's about the cell membrane, and we'll talk about different types of transportation like active and the passive, and we're going to talk about cell potential, like how the ions move from inside to the outside compartment in the cell in my next lecture. Thank you for watching. Bye-bye.
Please subscribe to see more videos regarding anatomy and physiology.